Um, so I apologize if for the people who have not been able to actually watch the class. So a biomaterial is a material that is intended to interface with biological systems to evaluate, treat, augment, or replace any tissue, organ, or function in the body. So some very important parts is this term biological systems, because something that material scientists don't need to consider that biomaterialists do is the fact that there is a biological system. So say that there's tissue, there's cells, any implant is a biomaterial problem, but it's not a material scientist problem. So evaluate, treat, augment, or replace any tissue, organ, or function of the body. So like stents or like even evaluation. I can't think of one of a biomaterial where you would evaluate something. But they interface with the biological system. Now, biomaterials is a very interdisciplinary field. And it's very, very important. So biomaterials is kind of this intersection of basic sciences like physics, chemistry, and biology, as well as medicine, because there's this translational part of it to where you use biomaterials in the clinic and in medicine, as well as engineering, because they serve a very specific purpose and it requires very specific solutions for it to be usable in medicine. And so the translation of biomaterials to actually become clinically relevant as a medical device, it depends on sound engineering design. So it has to have solid design, and you need a reason for every single thing that you do, for every single input that you put on your biomaterial. It also has to go through stringent testing in vitro and also in vivo. And so in vitro means it's usually out of the body, so like using cells or like a chip, like organ on a chip. And then in vivo is in a body, so like usually you'll start in mice, and then maybe you'll move up to pigs if you have the money, and then you'll see clinical trials. In terms of clinical realities, there has to be relevance. So even if something is feasible, if something is relatively feasible, but it just isn't very good, then that does not match that clinical reality. So, for example, making a implant to target obesity. So one of the very big, very effective treatments for obesity is a gastric bypass. Now, it is super, super effective. So even if you make an implant, which is maybe not as an invasive, but it's still very invasive, it's got to live up to the standard that gastric bypass has put. And so in that sense, an implant for obesity does not have that clinical reality and probably will not make it to market. And then the last point that makes biomaterials very clinically important is the involvement of industry permitting product development and commercialization. So there's the FDA waterfall that you have to go through, a lot of testing, a lot of documentation, as well as, a lot of money has to go into making a product to market. And that's the industry part. You need to almost have a company to be able to get a product to market. And a very famous, very prominent biomaterialist, Robert Langer, he's got like 4,000 different companies, and maybe half of them or more have to do with drug delivery and biomaterials. And so there is a COVID vaccine that's being delivered, and I forgot which one, but it is the company that is making it is one of Bob Langer's companies. So that's something that's kind of interesting. And then there's this complication of biocompatibility, which is the ability of the material to perform with an appropriate host response for a specific application. So something is biocompatible if it elicits an appropriate host response. So something that is inappropriate would be like it causes infection. Inflammation is a very touchy subject because sometimes you do want inflammation. Sometimes you do want to recruit immune cells to that area. So in my research, I do want some sort of inflammation and immune response because I do utilize immune cells for my therapy. And so in that sense, inflammation is desired for my therapy. But in a lot of therapies, it is not desired. So it depends on your application. And so a small kind of history of biomaterials is it kind of went in 3 different waves. In the first generation, which is, pre-60s, where their biomaterials wasn't really even a area in research. People were using materials that weren't intended or specifically designed for medical use, things like stainless steel. But they were selected because it had a desirable combination of physical properties. So they either don't degrade very much, or they have the right amount of stiffness, and they were also bioinert. And bioinert means that bioinert indicates that it induces a minimal response from host tissues. So stainless steel is a very good example. Silicone kind of came a bit later, but that is another example of a biomaterial that is very bioinert. And when the material itself came out, it wasn't intended or made for medical use, but it worked really well. And so examples would be elastomeric polymers as well as silicone. And then came the second generation of biomaterials where they actually are trying to induce a controlled reaction. And so they were bioactive. And so you're trying to induce a desired therapeutic effect. And so big examples of this include bioresorbable materials as well as in pharmaceutics using biomaterials for controlled drug delivery. And a big area for this are glasses and ceramics for dental and orthopedic surgeries. If you get a cavity and you get it filled, that ceramic counts as a bioactive material because it has to bond to your tooth, and it also has to be sterile. I don't know very much about dental and orthopedic surgeries, but that is one example. That was one of the earlier second generation biomaterials. And then the third big generation is more recent, where they're actually trying to create functional tissue regeneration and engineering. And so I kind of put a definition of tissue engineering where it's the interface of biomedical and engineering sciences that uses living cells or attract existing cells to aid in tissue formation or regeneration to produce a therapeutic or diagnostic benefit. And so an example of this would be to create a scaffold to make a functional organ or functional tissue. That would be a very solid example of a third generation biomaterial. So these are some examples of drug delivery biomaterials. So this one here, these are hormonal contraceptives. They are synthetic polymers that release hormones over time so that women don't get pregnant. So NuvaRing is one example, and then Mirena is a hormonal contraceptive that is also very popular. This is Eluvia. It is a drug-eluting stent because if you get plaquey arteries, you can get a stent implanted, but then sometimes you want their to be a more active response than just, widening the artery. You can actually get drug-eluting stents that release a drug over time to actually break down the plaque. And you can put in other factors too. This down here on the bottom is a flu vaccine, but it's a microneedle patch for the flu vaccine. This has been quite recent. I believe it was put into clinical trials just in the last several years, so I don't think it's like commercially available yet, but it saw a lot of promise in the clinical trials, so maybe we'll see that in a few years. This, I feel like a lot of teenagers now have seen, is a hydrocolloid patch for like acne and pimples, and that is a biomaterial drug delivery example. Hydrocolloid is a very interesting biomaterial to work with, and they usually put stuff in the hydrocolloid that attracts the pus. I don't know if it's actually pus, but that is also another example. So I grabbed this from a mRNA vaccine. It uses a liposome. So a liposome is a membrane. Words are failing me. It uses a membrane, like a lipid membrane, to, oops, Huey has jumped on the desk. It uses a lipid membrane to encapsulate the payload, and so it will include the antigen, or any mRNA or DNA. You can deliver a lot of stuff with liposomes, but there are a lot of vaccines that utilize biomaterial methods to deliver the payload. And then if you ever see anything that's like extended release, that uses a biomaterial as well. So I thought that would be kind of interesting. So I thought that it would be easier to go through this course by learning the drug delivery side before the biomaterial side. So I'm going to jump straight into drug delivery. And so the first part of drug delivery is fundamentals of pharmacology and pharmacokinetics. So here is where I'm going to try to make this interactive. So what would you see to deliver as a therapeutic? Can we boost this server to get, oh. Y'all can unmute yourself. Or I could just keep going. Yeah, so like would that be a protein? So we could put protein. Because proteins don't, well, some proteins don't normally cross the blood-brain barrier. Neither do cells. Sometimes you can deliver cells. You can deliver compounds, synthetic or even natural, synthetic and natural compounds. What else would you want to deliver? Radiation. Drugs. So not all of these would maybe be considered a drug. So a better term for drug delivery is honestly would be bioactive agents, because it's a broader term that encompasses things that are delivered as therapeutics but not considered drugs. And so a very good example would be radiation, because I don't think radiation is commonly thought of as a drug, but it might be considered a bioactive agent. And so the big goal in drug delivery, or bioactive agent delivery is to achieve an optimal concentration at a desired location for desired duration. And so these are three words that will come up a lot in this lecture. And so there are three main release systems. If you don't consider the bolus, which is just a one-time big dose, let it drop off slowly. And so an example of a bolus would be like an IV injection. And so in a controlled release system, the agent is not delivered in a single immediate dose. And so these are all considered controlled release. Now, what differs targeted release to controlled release is that targeted release, you are controlling the concentration of your agent in respect to the location. So say you wanted to target a tumor. That is a targeted release where it releases a payload at the tumor. And then there's a responsive release where it delivers a payload in response to the stimulus. And so a very example that I can think of is if you have a hydrogel and you have insulin in it. They'll just be little circles for now. But there's also glucose oxidase. Oops. And you could have this hydrogel to release insulin in the presence of glucose. And that would be an example of a responsive release. And so the reason why release systems are very important is because there's this idea of the therapeutic effect. Now, in most cases, you'll have the toxic effect be much higher than the therapeutic effect. And then down here, there's no effect. So you want to stay within this box for as long as you want and not go over to this toxic area where you'll see side effects. So most drugs are this way. Like if you take, if say you have a headache, you take some ibuprofen, maybe a baby dose isn't going to be enough for you, but then if you take like 2 pills, it'll do something, and that's good. You're in that therapeutic effect. But then if you take like 7, then you might see some toxic effects. But then there are some drugs where the therapeutic effect is above the toxic effect. And then down here, there's no effect. And so there is this balance that you have to consider of whether, these toxic effects are worth getting to the therapeutic effects. And an area where this is very prominent is in chemotherapy because, I think it's very well known that many chemotherapies have a very large toxic effect, but you also have to consider whether the quality of life is worth getting to this therapeutic effect. So you're trying to get a net benefit. So some important considerations you have to consider is like the desired concentration. So that is where, this comes into play. What concentration do you get to the therapeutic effect and not into the toxic effect? Then distribution is also very important, like whether it'll be distributed to everywhere in the body or like will it be only isolated to certain parts, and then how well does it retain in the body. So I mean, if you have to go in for an IV dose every 2 weeks, that is very different from going in for IV every day. And that's where, this could become a biomaterial solution. Actually, all of these can become biomaterial solutions, and I can talk about those later. But also quality of life. So quality of life indicates, how easy is it to take this drug to where you get the therapeutic effect. So if taking a pill is much easier than doing an injection. So whoever does if they ever find an oral solution for insulin, even if the cost is much higher, because the quality of life is worth it, there will still be a lot of people who would pay for an oral insulin dose so that they wouldn't have to take injections all the time. And so what is the difference between, apart from, route of delivery, what is an example of how a dose that is injected is different from an oral administration? So we have plasma concentration on this y-axis and time on this x-axis. If you administer at the same time, if you put something in through a vein, the plasma concentration will look something like this, where this is a bolus, where you get the full dose at one time, and then it drops off as either it gets absorbed, or it gets excreted, or it gets used. Now, an oral dose, you can get many different things. You can get, usually there is a lag time. Usually there's a lag time to when it goes into the body, and it will never be as high as the bolus, because some of it will just get peed or pooped out. Some of it, or it could not enter the body at all and just stay on that x-axis. So how it is entering the body is very important. So let's see, can I clear this one? How do I clear the screen? Apparently I can't. So say this is insulin. We'll go back to the insulin problem. If you inject it, you'll get this nice bolus response. But then if you take it orally, it's not even going to enter your body, because as a protein, it'll just degrade. So it'll stay on this x-axis. And this is why you have to inject insulin rather than take a pill. Let's see. And so because for injection, you're going straight into the bloodstream, you don't have to consider the absorption part of ADME. And I'll kind of explain what ADME is in a little bit. But there is like the distribution of magnitude and elimination issue for injected doses, because even though you inject a drug dose, it might not get distributed to the right parts of the body, or there might be too much or too little getting there, or it could just be eliminated by the body really quickly. However, for oral deliveries, you have to consider all of these. So like in the case of, for insulin, you had no absorption if you take it orally. So if you don't even get absorption, then how do you even consider distribution, magnitude, or elimination? But if it does get absorbed by the body, then you do still need to consider these. So in terms of plasma concentration, you'll get a much better, you'll get 100% of it entering the plasma, but sometimes you don't want this huge spike, because what if this was the toxic concentration? Then that is not desired. And if this line stays right below it, then maybe an oral solution is better. There are a lot of considerations in drug delivery. Okay, so I mentioned ADME before, and it is one of the 5 fundamentals of pharmacology and pharmacokinetics, and I'll talk about all of these things. ADME stands for absorption, absorption, distribution, magnitude, and elimination. And that is like how you can figure out how much you need. There's also the concept of site of action versus mechanism of action. Then you also have to consider biological barriers as well as routes of administration. And so what happens when you have a free drug. So ADME are the important rates of drug delivery. So you need to consider the rate of absorption. How how quickly does the drug absorb into the body? And so that is this line. And so if you consider that this free drug, if the free drug is in the plasma, does it even absorb into the body? And so this is determined by a rate of K or whatever. It depends on how it is absorbed, if it's passive transport or active transport. It depends. And then there is the rate of distribution or retention. And so this is determined by how it is distributed in the body. So for most agents, it will be distributed through the blood system, so like plasma. For something that's injected intramuscularly, it has to diffuse through tissue first to get into the systemic blood circulation. And this can decrease the rate of distribution. Or if you do like a subcutaneous injection, it has to traverse the fat layer and then the intramuscular layer before it can get through the blood circulation. Distribution is also affected by how long it can stay in the intended area. So a lot of drugs don't have their site of action in the bloodstream, and they have to target tissue effects. So how can the drug stay in the tissue without leaving it? And so the free drug, it can activate the receptor at the site of action. So say if the site of action is in the liver, then it is no longer free drug as it is bound to whatever receptor it's a ligand for. However, the ligand or the drug can also pop off of the receptor and be inputted back into the system as a free drug. So vitamin A is a very good example. You can ingest vitamin A and you'll get a plasma concentration of it, but it gets sequestered in the liver and that's where it's stored. And so as time goes on, and you're a bit low in vitamin A, your liver will also release vitamin A, and so that becomes free drug. Now, the rate or magnitude of a dose is how much should be given, and this question has a lot to do with the other rates, because you can't give too much or too little, as we've seen on the chart with the therapeutic effect, and so the dose needs to match the therapeutic effect intended. And something that I forgot to mention is that free drugs can also be sequestered at other locations. And so an example of that is in the plasma, in the plasma, it can be bound to other proteins. And so when it's bound to a protein like that, it is not available as a free drug. And so other locations can act as reservoirs. And then when it's needed, it can pop back into the blood as a free drug. And then there is elimination, how it exits the body or how it is removed as a free drug. And so there is a difference between excretion and biotransformation. Those are the main ways that a free drug will be eliminated from the body. In excretion, it is being removed without changing. And so example of this, if it's just like peed out in its bioactive form, then that is excretion. But then there's also biotransformation, where there's a chemical reaction to change its structure to where it is no longer the same free drug. And that is biotransformation. And so sometimes you can call it metabolism, but in a lot of cases, in some cases, it doesn't really count, and biotransformation is the preferred term to metabolism because sometimes there's no energy production involved. So say the conversion of hemoglobin to bilirubin is metabolism because I think there is energy production involved in that recycling. but for a lot of small molecules, especially ones that are not generated by the body, there's no metabolism involved. Alcohol is a very good example of where it is metabolic. That would be counted. That would count as a metabolic elimination method. But then something like insulin doesn't. It just gets degraded. Okay, so the next set of terms is site of action and mechanism of action. So the site of action is the location where the agent initiates the sequence of events leading to a very specific effect. And how you define that effect is very important. And then there's also mechanism of action, which is the means by which an agent produces what is defined as the effect. And so an example that I thought of is the contraction and dilation of the pupil, which is controlled by muscles in the iris. And so the mechanism of, in the case of morphine, its site of action is the CNS, so the central nervous system, and its intended effect is contraction. So if you put it through an eye drop, if you put morphine on the eye in the form of an eye drop, then there's no effect. However, if you put it through an IV, then you get contraction. And in this case, the site of action, because the site of action is the central nervous system, it can induce its mechanism of action, which is contraction of the muscles in the eye. Now, atropine has its site of action in the eye muscles itself. And so if you give it IV, then there's no effect. Because the eye has very regulated transport of what goes in and out of that barrier. However, if you put it through an eye drop, then you get dilation. So in this case, the site of action, which is the eye muscles, are able to receive the signal from atropine, which induces the eye muscles to dilate. And so that's where site of action and mechanism of action come into play. And so the desired effect also differs. So in the case of morphine, the intended effect is for contraction. And for atropine, the intended effect is dilation. So how you define the effect is very important in drug delivery. All right, so I was going to go into a bit more depth as to what site of action is. And so you can have extracellular and intracellular sites of action. So extracellular sites of action are usually very simple. They have low specificity, and there usually isn't very much interaction. And so a very good example is an antacid. So an antacid works by NaCO3 plus HCl to H2O. Please excuse my ugly writing. Cl and CO2. So how an antacid works is it just reduces the acidity in the stomach by a simple chemical reaction. It reacts with the acids in the stomach and converts to water and salt and carbon dioxide. So that's an example of an extracellular site of action. And then you have intracellular sites of action, which are much more specialized. So you have specialized macromolecules. This could be a protein. It's usually a protein that's able to recognize agents with a lot of selectivity to trigger a biological response. And this biological response can be a signaling cascade. And so these molecules are usually receptor and ligands. And there are a couple of interactions between a ligand and receptor that occur. So you get covalent bonds, if you remember from like high school chemistry. You can get covalent bonds, which are very strong, but they're also not reversible. And you do have drugs that, you do have ligands that will initiate a covalent bond with a receptor that doesn't pop off. So like it is permanently there until it's just degraded. Then you also get ionic bonds, which are strong and reversible. You get van der Waals, which are very, very weak, and they have to be very close together for it to really interact. Usually it's not the initial interaction. Usually the other ones like covalent, ionic, hydrogen, or hydrophobic bonds are the main ones, and then van der Waals happens as an afterthought. Hydrogen bonding is very, very important in biology. There's hydrogen bonding in like DNA. There's hydrogen bonding in many, many proteins. So this is a very prominent one. And then there's also hydrophobic bonds, which I think a lot of people don't really think about. But most of the times, hydrophobic regions in like a protein are usually hidden, but they're still very relevant. And they can initiate some very interesting chemistries, especially in biomaterials, and they induce some really interesting things in biology. So an example of hydrophobic interactions would be like collagen. Normally collagen, if you like don't put any strain on it, it like coils up because it has hydrophobic regions, and then when you put energy into it, then it like stretches out. Well, it's usually a wiggle. So that's where hydrophobic bonds are really important. And so just to drive this point home, for a site of action, this is acetylcholine. And so a question that I want to ask is like, how can we form the strongest receptor for acetylcholine? So how can we make something that's very, very specific, and we know that it's going to initiate some sort of response? And so the answer to that is to maximize the type and as well as number of bonds. So if you see here, it has a couple of oxygens where you can initiate H bonding. However, oops, I spelled that wrong. However, the really big one is this amine. Because with this amine, you not only get hydrogen bonding, but you also get ionic bonding. And that's a really big one for ligand and receptors. And then you can also do van der Waals. Now, how van der Waals comes into play is that if you maximize the surface area of the interaction, then van der Waals can be very, very strong. So if you have, like, say, if your receptor just looks like this, where there isn't very much area for the ligand and receptor to interact, then your van der Waals bonding is quite weak. But if you have something that looks like this, then your surface area is much larger, and then your ligand or whatever it is has a lot of area to where it can interact, and there's much more van der Waals forces going on. And so this is where the 3D conformation is very important for ligand and receptor bonding. So I also want you to note that, there are cellular sites of action that don't have receptor. A very prominent example are detergents, which disrupt the cell membrane, as well as DNA interactions are also count as cellular sites of action, but don't have receptor. And so an example would be chemotherapeutics that interact with the DNA. Those don't have a receptor, but they still have a site of action on the DNA molecule. How am I on time? I have 10 minutes. So mechanism of action also has an extracellular and an intracellular component. So example of an extracellular component would be heparin. And heparin works as an anticoagulant because it inhibits the conversion of prothrombin to thrombin. And thrombin is necessary to convert fibrinogen to fibrin, and that forms the clot. So that's how heparin works, is that it's extracellular, prevents conversion of proteins that are required for clots to form. So intracellular examples would be pepsid, which has an antacid, which is extracellular, but there's also famotidine, which inhibits histamine receptors on parietal cells on the stomach lining. So this is on the stomach. And what it does is it reduces the amount of hydrogen available by inhibiting this hydrogen pump. So this is what famotidine does. That's what famotidine does, is that it inhibits this hydrogen pump so that hydrogen can't enter the parietal cells to be used to be pumped into the stomach. Stomach is on this side. Prilosec is another intracellular mechanism of action, and what it does, Prilosec inhibits this pump, the potassium hydrogen pump, and so that's how it also increases the pH in the stomach by preventing hydrogen from entering the stomach. Now, Taxol has nothing to do with the stomach. It is a chemotherapeutic, and it stabilizes microtubules to interfere with mitosis. And so it targets proliferating cells and BCL2 as a cancer therapeutic. And so that's how Taxol works, is that it prevents mitosis from occurring as well. It also prevents meiosis from occurring, and that's how it prevents like the continual replication of tumor cells. And I think this would be a good place to stop because it is 6:54. And so are there any questions? You're welcome to unmute yourself, and I can go back and forth on this PowerPoint. Oh, bolus? So a bolus, So a bolus means that you are putting in the dose all at once into the blood. So an IV is a very good example of that. I can't really think of any other method that it would be a bolus. I guess if you put something into the peritoneal cavity, that could be a bolus. So it doesn't have to be the blood. Like you could have a bolus injection into the heart with epinephrine, which is adrenaline, and that's a bolus. Yes. Biochem. I will be not talking about any biochem because I am not good at biochem. Also, I don't know if Dad Patrol is. Oh wait, no. Dad Patrol just asked a question, but not related to the lecture. Are there any other questions or any comments? Y'all are so quiet. Alright, if there are no more questions, so I should be able to do lecture again next week. We'll see how my schedule goes because it's kind of fucky. Which it is every week, but it was a good question this week. Lolly, do you have any questions? You can also type it in the chat. I've got 3 screens going, so I see everything. Okay. Well, anyway, I know Lolly has her lecture this weekend, so be sure to watch that one. And I guess now a lot of people know that I am actually a girl. So I think most people assume that I'm a guy, but I am indeed gal. But thank you for coming to my lecture, and I will post the recording on Biocord as soon as I figure out how. Well, thank you for coming. Thank you. How do I stop sharing?